Hello everyone, welcome to today's webinar. I'm Kelly McSweeney, contributing editor at CNN Media, and I will be moderating today's event. This webinar is titled Recent Advances in Library Synthesis to Accelerate MedChem Research and is being sponsored by WuXi AppTec. CNN works with sponsors to identify topics of interest and value to CNEN's audience, and consistent with CNEN's mission to provide news and analysis of the chemistry enterprise in a timely, accurate, and balanced fashion. During the webinar, you can adjust the size of the slides on your screen by grabbing the lower right corner with your mouse. If you need technical assistance, please look at the Help widget at the bottom of the screen or type your query into the Q&A box. If you are disconnected during the webcast, please log in again according to the instructions you received earlier. You are encouraged to contribute to the success of this webinar by asking questions at any time during the presentation through the Q&A box on your screen. The questions will be answered at the end of the presentation, and as your moderator, I will be posing as many as time permits. Please note that CNN does not endorse any company, products, or services that may be mentioned in the webinars, and that each webinar will be archived at CNN Online after the live webcast. The presentation today is being sponsored by WuXi AppTec. To introduce WuXi AppTec, we will hear from Dr. Declan Ryan, Vice President of the Research Service Division. And I will now hand it over to Declan.
Thank you. Welcome, everybody. I'd like to share a few words about WuXi AppTec. WuXi AppTec provides a broad portfolio of R&D and manufacturing services that enable companies in the pharmaceutical, biotech, and medical device industries worldwide to advance discoveries and deliver groundbreaking treatments to patients. With industry-leading capabilities such as R&D and manufacturing for small molecules, cell and gene therapy, and testing for medical devices, WuXi AppTec's open access platform is enabling more than 4,400 collaborators from over 30 countries to improve the health of those in need, and to realize the vision that every drug can be made and every disease can be treated. During this presentation, you will hear from Dr. Yan Zhu. Dr. Zhu is Executive Director for the Chemistry Service Unit and has more than 20 years of experience in library synthesis at WuXi AppTec. She is especially experienced in the application of parallel medicinal chemistry model to accelerate early drug discovery projects towards lead optimization. She established high-throughput synthesis, purification, QC, and compound logistic workflow to enable fast parallel synthesis. Dr. Yan earned her PhD at Nanjing University. Yan, over to you.
Hi everyone, I'm Yan Xu, worked in WuXi AppTec since 2001 for library synthesis. Thanks a lot for your attendance for this webinar. It's my pleasure to share with you about recent advances for library platform and how parallel chemistry can accelerate drug discovery stage. I started my career in 2001, so almost witnessed the change and evolution of library in past 20 years. In the first part, I will introduce the changes and recent advances in library platform, which includes virtual library, library enrichment, parallel medicinal chemistry, and analytical PMC, Covalent library, and the PMC to accelerate PROTAC research. The most important thing for the pharmaceutical industry is to deliver drug candidates in a more cost-effective and timely manner. Thus, the concept of parallel synthesis and the combinatorial chemistry have been developed in the later part of the 20th century. Many of these campaigns focused on preparing thousands of compounds to enrich corporate compound collections and provide leads for targets emerging from human genomic projects. Nowadays, library synthesis has been widely and effectively applied to accelerate drug discovery in the stage of target to hit, hit to lead, and lead optimization. During the last 2 decades, WuXi AppTec has developed many enabling chemistries and high-throughput technologies to improve the access to expanded chemical diversity by library synthesis. WuXi has built up a highly productive library synthesis platform to champion the design, synthesis, and testing target molecules to accelerate drug discovery programs. WuXi Library Platform is part of WuXi Chemistry Platform, which can support chemistry projects from discovery to full development stage for both farmers and the biotechs. On average, an approved drug currently costs $2 to $3 billion US and takes more than 10 years to develop. In part, this is due to expensive and time-consuming wet laboratory experiments, poor initial heat compounds, and high attrition rate in the preclinical phases. Structure-based virtual screening has the potential to mitigate these problems. We have developed the WuXi Virtual Space and working with Bisolve IT, the software company, to build a very fast searching tool, Galaxy. So far, there are 2.3 billion virtual compounds in this space. And due to the part 2, which based on WuXi unique templates and building blocks, made our virtual space only have 10% overlap with others. The compounds are very diverse and have chemistry feasibility, which could be delivered within 4 to 6 weeks after compound selected. There is also plan to build the space to 10 billion scale in the year of 2022. It is well known scaffolds containing oxidizer moiety are very important, of course, for drug or drug candidates. In our virtual space, there are more than 6.5 million oxidizer ring compounds designed based on in-house parallel strategy, which are very attractive for many medicinal chemists. However, despite the fact that large database of compound exists, the ability to carry out large-scale structure-based virtual screening on computer clusters in an accessible, efficient, and flexible manner has remained difficult. Worked with Bisolve IT, a very fast searching tool, Galaxy, was established. Taking BTK inhibitor as an example, the compound structure could be pasted here, and you can set different criteria by similarity or diversity. Then only in 1 or 2 minutes, you can get a searching result by thousands of structures. You can easily save the virtual heat structure as SD file, and mostly the new chemotype could be found as a starting point. The Galaxy tool and the SD file of enumerated virtual compounds could be accessed by the linkage showed at the bottom of this page. In 2020, our virtual space also supported his finding project for COVID-19. Our client carried out virtual screening against some of the published SARS-CoV-2 protein target sites to get around 6,000 virtual hits. We synthesized around 600 compounds in high ranking score and delivered 3 copies for each compound within 5 weeks to their 3 partners. The test is still in progress now. Hopefully, there will be some active compounds identified as COVID-19 virus still spreading in many countries. Now, library enrichment is still used to support target-to-heat and heat-to-lead stage, but it also evolved a lot. Not like early days that thousands of compounds each library were made, designers aimed for new chemical space and more novel and diverse library. Only 200 to 400 compounds designed for each library, and even with multiple different cores. For the design scaffolds, almost require no heat found in the literature or patent. That means the chemistry rough coding becomes very challenging. And majority of the libraries need 8 to 10-step synthesis. WuXi also becomes a very important partner to generate library ideas based on 20-year experience on template design and synthesis. About 46% of the library proposals come from WuXi, which could be run either on the exclusive model or non-exclusive model. This also requires very comprehensive synthetic capability, which could be up to multiple kilogram scale synthesis for scaffold and parallel synthesis at 20-gram scale for key intermediates, and also milligram scale high throughput library production. In recent years, we prepared more than 1,000 scaffolds in around 800 gram each, 9,000 K intermediates in 10 gram each, and 250,000 library compounds in 30 mg each. Quality control results show the compounds are all in very high quality. The 1990s witnessed the emergence of combinatorial chemistry and the synthesis of large compound arrays as screening library. However, in 2000s, the art and science of combinatorial chemistry has also undergone paradigm shifts from the initial emphasis on large combinatorial matrix to small focused library. And also many innovative capabilities developed and applied to make better molecular faster. This also makes demand of large library declined. Meanwhile, parallel medicinal chemistry is routinely applied to accelerate lead optimization stage. The main purpose of PMC is to shorten turnaround time of design synthesis test cycle and provide more diverse and novel compounds with limited scaffold by streamlined workflow. The high speed, high success rate, and the more diverse reaction types, as well as flexible adoption of new technologies such as catalyst screening, photoredox chemistry, microscale synthesis, and high throughput purification will help medicinal chemists to do lead optimization in a fast manner. Here is an example how PMC library can accelerate the cycle of design synthesis test. With accumulated parallel synthesis knowledge, these 5-step libraries could be handled in parallel format even from the first step. Each cycle focused on optimize one site for the molecular, could be executed within 2 to 3 weeks and in 75 to 100% success rate. Thus, within 6 months, 6 cycles have been carried out and lead compounds have been found. While by traditional Medicam analog synthesis, only 2 to 3 cycles could be run and the synthesis cost will be much higher. Parallel medicinal chemistry started from 2006. After 15 years continuous improvement on workflow and by accumulated parallel synthesis knowledge, cycle time has been shortened to 2 to 3 weeks with increased synthetic success rate, now reached to average 85%, which means with same amount of material, we could provide more compounds much faster to medicinal chemists for their testing. Although PMC library can provide compounds very quickly for lead optimization, sometimes the headaches for medicinal chemists at the scaffold are very precious, and at the beginning they don't know which direction to go, so they want more primary pharmacology data to make decisions, means they need more compounds to get more data. To solve this problem, analytical scale PMC was developed to carry out the reaction in 10 to 20 micromole scale, usually 3 to 5 mg each reaction. And by more rapid purification and dry down workflow, with the same amount of scaffolds, it can give number of compounds 5 times more than by normal PMC. The amount of product is 0.4 to 3 mg each, which is already enough to get primary pharmacology data. Once active compound is found, it is easy to come back to do resynthesis with established reaction conditions very quickly. Here are several examples to show with 300 to 500 mg of scaffolds. By analytical PMC, it can provide 60 to 70 pure compounds within 8 to 9 days. Different apparatus such as parallel reactors for sealed plate reaction, glove box, kits were used to make sure the success for such smaller scale reaction in parallel. Multiple steps library also could be handled on the analytical PMC model. With the same amount of the scaffolds, by analytical PMC, it could generate 5 times more compounds than by PMC. The second case showed with 600 mg of scaffold, 144 compounds were obtained, while normal PMC only can provide 28 compounds. However, if medicinal chemists need more amount of the active compound to run more biology assays, it is easy to do resynthesis on the normal PMC model. So combination of analytical PMC and normal PMC, it can even move faster for lead optimization. In the following, we will see how PMC can accelerate the research of some hot areas for drug discovery. Covalent drugs have experienced a significant renewed interest in drug discovery. This resurgence has been accompanied by a better understanding of the reactivity relationships required to engage selective covalent bonds between nucleophilic proteins and electrophilic small molecules. Recently, Amgen discovered and developed a covalent inhibitor of KRAS G12C, AMG 510, which makes covalent small molecules a very hot topic in drug discovery within the last few years. A lot of efforts have been made by either biotechs or farmers to find highly potent and selective covalent inhibitors. Library synthesis has been a very useful tool to support medicinal chemistry research employing covalent chemistries. Generally, covalent warheads serve as electrophile which react with nucleophiles such as cysteine thiol in the biological system to form covalent bond resulting in superior reactivity. Cysteine-targeted covalent chemistry has been indisputably fruitful for delivering safe and relatively selective drugs. Nowadays, a lot of novel warheads are rapidly emerging within the scientific literature, aiming for expanding the toolkit of reactive modalities that are amenable to chemical biology and drug discovery. Picture on the left shows the selected examples of electrophiles known in the literature to target nucleophilic amino acids. From top to bottom, it is from well-established to increasing novelty. At the right side, these are warheads we have validated for cysteine reactive, lysine reactive, and histidine reactive. The chemistry has been well established. Here are several cases that using library tool, it can convert hundreds of nucleophile molecules to covalent compounds within 2 to 4 weeks by high success rates. Also, expanding the repertoire of electrophiles with unique reactivity features would facilitate the development of covalent inhibitors with the desirable reactivity profiles. The picture at the right side shows the bicyclobutane carboxylic amide as a new class of cytoreactive electrophiles for selective and irreversible inhibition of targeted protein. By tuning BCB amide reactivity and optimizing its disposition on the ligand, the authors obtain a selective covalent inhibitor of BTK. Continue following the literature closely and validate and recommend new warheads to medicinal chemists. Another hot area, PROTAC. The advantages of PROTAC are well known as it can degrade target proteins specifically with selective binders. It targets undruggable proteins and requires low dose or concentration and prolongs the duration of action. However, chemistry difficulties, compound stability issue, purification challenge, and long synthetic cycle time also are the challenges for PROTAC research. Here we use several cases to demonstrate how PMC could accelerate PROTAC research. The first case is the modification for E3 ligase ligand. Different synthetic strategies such as parallel microwave reactions and photoredox chemistry were used to produce 228 different E3 ligase ligands within 2 to 3 weeks with 76% success rate. Different E3 ligase ligands could be done diversitization in parallel format, thus increase the speed for PROTAC research a lot. The second case is using parallel Medicinal reaction, then by oxidation to prepare 50 different linkers, then to form PROTAC compounds. The work of total 200 synthetic steps and high-speed purification and QC were all done within 4 weeks. We can handle different types of linker sterilization in parallel formats and then assemble to form PROTAC compounds within few weeks. The third case is using 57 different linkers and the 2 E3 ligase ligands to generate 97 PROTAC molecules within 6 weeks by 228 steps with 85% success rate. We are still investigating and applying parallel synthesis strategy into other hot areas for drug discovery and try to further speed up the lead optimization stage in more cost-effective way. Hopefully, we can share more progress in near future. Then, in the second part, I'd like to introduce what are the critical factors for PMC. This also demonstrates how we optimize PMC process to speed up the Medichem research. The whole process includes 4 stages: reagent acquisition, synthesis including chemistry validation and library production, then purification and QC, and finally compound management to prepare acid-ready plate or transfer solid to preferred vials. The first stage, reagent acquisition, also takes 1 week, which is already 25 to 30% of the total cycle time. So reagent acquisition speed is very critical to the speed of PMC library. In WuXi, the warehouse stored more than 37,000 monomers, which could be obtained within the same day or the second day. The storage list was often asked by medicinal chemists to use it to design their PMC library. Furthermore, WuXi Lab Network also can do procurement for more than 220,000 reagents within 3 to 4 days, which ensure the quick start of the PMC library. We also provide a service that manages the monomers synthesized or purchased for clients exclusively to further reduce the monomer acquisition cycle time. For example, for one collaboration, we managed more than 6,000 building blocks for clients, and around 4,200 building blocks, which about 70% of the total were reused for library synthesis for past 5 years. By this model, it not only reduced the cycle time by 33%, as majority of the building blocks are in storage, but also reduced the cost of whole library by 35%. Then the second stage for PMC is synthesis, which includes chemistry validation and library production. More than 46 reaction types are widely used for PMC to generate diverse compounds. Protocols were well developed to run some reaction types which seldom applied in parallel synthesis previously. For example, by using solid-supported reagents, such as polymer-supported triphenylphosphine, it can enable Wittig reaction in parallel format. By palladium catalyst and ligand, the carbonylation also could be used for library synthesis. By Bestman or Hero reagents, it can in situ generate alkene from aldehyde parallelly, then used for click chemistry, which expanded the compound diversity. More cyclization reactions showed here to form drug-like cores in parallel formats for Medichem projects. Recently, photoredox chemistry also used significantly in PMC library to reduce synthetic steps and do cross-coupling more efficiently and innovatively, which I will introduce later. We all know chemistry is chemistry. For library and the Medichem synthesis, it is commonly happen that one reaction condition works for some monomers, while other monomers cannot afford products. So kit screening is a very powerful tool to solve chemistry problems within 1 to 3 days. Our kit screening platform can screen conditions for 39 reaction types with around 70% success rate. Nowadays, condition screening also runs in microscale with less than 1 meg of samples for each condition. It can save precious material for medicinal chemists. Here is an example. The chemist made problem for backward reaction, then submit it to kit screening platform. 24 conditions with 6 different catalysts and 2 solvents and 2 bases were tried at 120 degrees. The product was formed in 70% by one condition, but atomization will also happen. Then second round of screening was carried out with 12 catalysts in 2 different solvents on the lower temperature at 40 degrees. By T-butyl export palladium third generation catalyst, it gave product in 70% yield without any atomization. This condition was used for library production successfully. The problem got to be solved within 3 days. Kit screening is a very powerful tool to solve the problem quickly, improve the success rate, reduce the cycle time, as well as develop a new protocol for both library and medicam synthesis. By accumulating experience, we have established more than 120 protocols for 16 often used reaction types. We keep on developing new protocols which will make us run PMC library even faster. Here is an example of protocol development. Click chemistry is a concept in which modular synthesis is used to rapidly find new molecules with desirable properties. But the availability of azide reagents is limited owing to their potential toxicity and the risk of explosion involved in their preparation. On the top, imidazole sulfonyl azide is often used to prepare organic azide compounds, but it's more suitable for alkylamines. For aromatic amines, sometimes it gives low yield. At the bottom, it shows the new protocol was developed to prepare more diverse azide compounds. This new protocol can cover a wider scope, not only suitable for alkylamines, but also for many aromatic amines. This can give higher yield than traditional methods. It's a very efficient protocol to extend azide diversity for click chemistry. The second case is the protocol for parallel thumbnail reaction. Heterocyclic array halides have limited availability compared to amines. So starting from amines, where parallel thumbnail reaction, it can increase the diversity of the molecule significantly. This condition is mild to handle in parallel format, and we continue to develop new protocols to enhance our PMC capability. Now I will introduce how to use photoredox chemistry for parallel synthesis. In 2013, Professor David McMillan published the article, Visible Light Photoredox Catalyst with Transition Metal Complexes Application in Organic Synthesis. It brought a lot of new ideas for organic synthesis route scouting and also brought many possibilities and faster approaches for PMC library design. We have established from microgram scale for photoredox condition screening capability to milligram scale PMC photoredox library capability to gram scale scaffold synthesis capability. Combined with flow chemistry, it also could be scaled up to even kilogram scale. Here are 2 examples to apply photoredox in PMC library synthesis. The left one is the carboxylative coupling reaction, which now widely used in the synthesis for medicam projects. The condition of this reaction is mild and can cover a wide substrate scope with high efficiency. And the medicinal chemists can use this for later stage functionalization on their drug-like course. We have different photoredox parallel reactors to run 100 reactions very easily. The case on the right is the carbon-carbon cross electrophile coupling The halide reagents are easily to get, and this reaction gave high success rate and reproducibility. While by traditional organic synthesis, it might need multiple steps. This gives medicinal chemists the freedom to do derivatization at final step. This case showed photoredox combined with flow chemistry to produce scaffold for library synthesis. In the literature, the batch reaction reported with very dilute reaction solution. It is very difficult to do scale-up by literature procedure. By the in-house assembled flow system on the blue LED light, the scaffold could be scaled up to 50 grams, which is enough for library synthesis. By this way, medicinal chemists can get more novel scaffolds very quickly for their research. We are still developing more photoredox protocols for PMC library now. Here comes to the third stage, purification and QC. The process was well established in high throughput way for both purification and quality control. Not only the QC results, but also the QC report will be provided along with compounds. Our QC results are highly consistent with client QC or third-party QC, which also demonstrate the compounds delivered are in high quality. Here comes the final stage of PMC, compound management. It includes generating solid copy or solution copy for different purposes, compound storage, and tracking. By previous model, we ship compounds to clients and then clients ship them to their assay partners. There are two shipping cycles. By the compound management model, the clients only need to give plating or shipping instructions. Then we can send the solid or solution samples to their assay partner directly, which can reduce 50% of the total logistics time. It is usually 7 days could be reduced. This shows we can collect and store compounds from different partners or collaborators and manage the compound in WuXi. By the instruction from clients, we can generate the assay-ready plate and ship to assay providers. This is to enable any assay anywhere. The tissue mosaic system or WuXi's compound tracking system could make the compound amount or compound status visible to clients exclusively. And we don't need the structure information. The only information we need are the molecular weight and salt form for the compound. If the chemistry partner and the assay partner are all in China, the cycle time from compound ready to start test is only 1 or 2 days, which significantly reduces the logistic cycle time at least 7 to 10 days. Here shows we can handle very sophisticated workflow for compound management. The solid copy could be used for storage, and the solution plate could be used to run assay immediately once client receives them. We passed client audit without any issue found. So far, by this, it is entire process for PMC library. As a summary, to effectively accelerate drug discovery in the stage of target to hit, hit to lead, and lead optimization, we have built virtual library capabilities, library enrichment capability, and the PMC capability to establish WuXi library platform. In the future, we will focus on utilizing robotics, automation, and artificial intelligence and other new technology to further enhance library capability to support drug discovery in more timely and cost-effective manner. At last, I would like to thank my colleagues, Dr. Yu Gao and Dr. Li Qiangqu, who also have more than 18 years library experience and contributed a lot for this presentation. Also appreciate my colleague Declan for his timely help and fully support for our work. Thanks a lot for your attention.
Thank you, Dr. Yan Xu and Declan Ryan, for that excellent presentation. And we will provide slides after this live webinar for anyone who would like to see them. We have some questions from the attendees. So the first question is, how do you strip the solvent after HPLC?
Okay, thank you for the question. We have a high throughput lyophilization instrument which can handle hundreds of the HPLC purification fractions. It is very efficient and can give a dry powder mostly for the sample. And it's in very high throughput and very efficient way.
All right, the next question is, how do you remove impurities from each sample in a parallel library?
Yeah, thanks for the question. So as I mentioned in the purification and QC session, we have set up a high throughput purification workflow and use a preparative HPLC, which could be detected by the UV and also by mass triggered HPLC for the purification. And after that, we will collect the fraction and identify which fraction is the right compound, and dry down the fraction and send the compound for QC. It is the whole process for purification and QC.
All right. The next question is, how are you dealing with inorganic bases in a UMOL scale? Slurry solution, solid dispenser?
Yeah. For the inorganic base, usually we buy the solid dispenser.
Okay. The next question is, how do you purify covalent library?
Yes. For covalent library, generally we also purify the final compound by the preparative HPLC. Indeed, for some warhead, it is more challenging than the normal library, but we have already established the purification method for the covalent compound. Thus, make the high recovery rate and also the high success rate.
The next question is, do you observe good reproducibility of results from analytical PMC to normal PMC? Are the reaction yields maintained?
Yes. So for the analytical PMC, it's run in a much smaller scale. Usually, the yield and the recovery rate is lower than the normal PRC, but it can produce the compound very quickly, and with limited scaffold, we can produce more compounds. Compared to the normal PMC, since the normal PMC usually carry out around 50 micromole to 100 micromole, the yield and the success rate is usually higher than analytical PMC. So the reproducibility is very good. And as I mentioned in the presentation, the combination of normal PRC and analytical PRC sometimes can even move the project faster.
The next question is, how do your workup reactions in parallel that contain inorganics before purifying?
Yes, we have a parallel SPE cartridge which can remove the inorganic impurities before we send for the HPLC purification. We have established the workflow, and it is quite efficient to do the pre-purification and make HPLC purification higher yield and high success rate.
What tool do you use to track library synthesis and batch processing?
We have our internal, we call the library tracking system. It is the internal developed tracking system by our IT guys. And it can track starting from the synthesis and then to the purification and the fraction dry down, and even the weighing process until the compound delivered to our client. So all the information are traceable, and every compound, even every fraction, will tracked by the 2D barcode.
The next question is, what does success rate mean? Is it that any percentage of product is formed? Is it 50%, greater than 90%?
Yes. Success rate, usually the compound after purification obtained means the pure product, and then compared to the number of reactions we carried out. So it is depend on the amount we obtained from the product. For example, if a client wants 5 mg, we calculate the success rate based on the number of compounds about 5 milligrams, and then compared to the number of reactions. Indeed, sometimes for the library synthesis, parallel synthesis, the yield is not very high, but even with 40% or 50% yield, after purification, we still can get the compound. And this could be still also be included in the successful cases. So it is calculated by the compound delivered versus the number of reactions.
How many people are working in the PMC group?
Currently, we have around 80 people working on the PMC group.
And the next question is, how do you set up the reaction screen with less than 1 milligram starting material? All reagent amounts presumably will be less than 1 milligram. Do you rely on stock solutions and slurries?
Yes, for the small scale, we usually use the solution to do the library setup. You know, as such a smaller scale, it is very hard to do like a solid transfer. So we usually dissolve the starting material in the solvent, and then by pipetting into the microtubes to do the reaction screening. So sometimes for the often used reagents, we also use the stock solution. And of course, we will make sure the stock solution in good quality to make sure the reproducibility of the screening results.
What is the range of wavelengths of light to be and or was used for photoreduction process in PMC?
Okay, so if the question is concerned about the photoredox reaction in the presentation, the light is a visible LED light. It's usually, if I remember correctly, around the 400 to 700 range.
Do you use stock solution of monomers for library synthesis?
Actually, no. Usually, before we prepare one library, we will prepare the solution freshly to make sure the quality of the solution of the monomer and to make sure the success rate of the library synthesis.
Could you provide some additional details on the reaction setup and reactors that you use?
Yes, I think for this question, we can discuss offline to provide the specific information about the parallel reactors. And we have different kinds of parallel reactors to support parallel medicinal chemistry and as well as analytical PMC.
Okay, the next question is, what tool do you use to track library synthesis and batch processing?
I think I answered that question before.
Okay. All right. We'll move on to the next question. The next question is, how do you predict the feasibility of PMC for a scaffold? Before the PMC, is it necessary to first try the classical chemistry for several compounds?
Yes. Usually after we get a new library, and we will firstly look through the reagents, the monomer list, and based on our experience, we will classify the monomer to different groups according to the reactivity or according to the function group or some other factors. And then from each group, we will select 2 to 3 representative monomers to do the chemistry validation. And if after the validation we get the suitable condition, we will use the condition for this group of the monomer. So that's sometimes we usually use several different protocols for one library due to the different reactivity of the monomer. And by this way, it can make sure the success rate for the whole library, and we can establish the different protocol for the whole library according to different groups of the monomers.
Okay. What are the biggest challenges faced when conducting PMC reactions?
Yeah, it's a very good question. I think the challenge currently is still we want to use more reaction type to further actually enlarge the compound diversity and the compound novelty. You know, in the parallel format, still a lot of reaction types cannot work very well in the parallel format. Currently, we can handle more than 46 types of reactions, but we still try to develop more protocols for more reaction types. Thus, we can actually generate more chemotypes and also provide a more diverse compound for the to the medicinal chemists.
Do you observe good reproducibility of results from analytical PMC to normal PMC? Are the reaction yields maintained?
Yes, it is. The reproducibility between the PMC and analytical PMC are very good. So that's the reason we think the combination of these two different models can even move the project faster.
You mentioned parallel reactors. Can you disclose what type and brand these parallel reactors are?
Mostly from the Chinese vendor. And also in this year, we are trying to develop our own reactor by the robot arm. Maybe in the near future, we can share some information about the robotic parallel synthesis reactor with you. So hopefully at that time, we can further improve the synthesis efficiency for the parallel chemistry.
How is analytical data such as 1H, NMR, LCMS of PMC reactions analyzed for large sets of products?
Yeah, usually every compound will be checked by LCMS to make sure the right molecular weight and also the purity. And sometimes the client also ask for 10 to 20% of the NMR data to verify the structure of some representative compounds. So we also can, we're also very flexible for the QC criteria according to the client's demand. It is or could be discussed for different QC criteria.
Do you have a reagent score database for each type of chemistry?
Yeah, we have actually gave the monomer, the commercial available monomers, a kind of a score. We call the monomer performance, which often gives success rate will have a higher score, while the low yield or low success rate have a lower score. So it is also a very good knowledge for the PMC synthesis.
Okay, the next question is, how are the analytical PMC products characterized? Only LCMS or also NMR? Are the solids also dried down and weighed on the scale?
Yes, for the analytical PMC, usually it is characterized by LC mass. Since the amount was only 0.4 to 3 mg, it is hard to check by NMR. The solid, of course, is dried down and weighed to the microtubes to prepare the 10 micromole solution for the assay testing. The whole process actually, we already done the quantification to make sure the concentration are accurate. Thus, the biology data is reliable according to the whole process.
Do you often come across difficult to handle products when performing PMC? On several occasions, I've had products crash out of solution, which I imagine could hinder a universally applied workflow when applied to a wide range of substrates.
Yes. Sometimes we also found the compound that crashed out from the solution, especially for the low solubility compounds. And for this, I think it is very critical when you do the validation. You need to actually observe the reaction mixture very carefully. And once this happens, we actually will pay a lot of attention whether the product will crash out from the solution, and then we will do the filtration. And also later, maybe we don't apply the general workflow. Instead, we're doing the micro scale recrystallization to get the pure product. And this is more efficient, actually, than the HPLC purification for such low solubility compound.
What are the % yields from this procedure?
Yeah, I'm not sure this question is asking for which procedure. I think generally for the protocol we developed, usually the yield actually average from 40 to 80% cover all kinds of monomers. So even lower than like 30 or 40% of the yield, we still can do the purification and get some amount of the product.
How do you purify PROTAC library?
Yes, purification of PROTAC library is indeed a very challenge. We have established different methods because we already have more than 5 years experience in this area. We developed the protocol for both synthesis and high purification. And we are capable of carrying out the multiple step synthesis. And also, we have developed more than 200 different purification conditions, including different columns, modifiers, and gradients. And in our company, the analytical group, there are dedicated teams working on the purification for PROTAC compounds. It's usually by protective HPLC, and we already purified more than 50,000 PROTAC molecules. So if you would like to know more about this capability, please feel free to contact us. We can share more information offline. Thank you.
Okay, the next question is, do you make stability tests of compounds of a covalent binder library, and what experiences have you made?
Yes, indeed. So for some warhead, actually the compound is not very stable. So if we find the stability issue during the validation, we will carry out the stability test. There are protocols for the stability testing which are agreed with our clients, and we will do the stability testing according to the protocol and provide the full data to the client if we find the stability issue.
Do you have enantiomer-rich libraries?
No.
Okay, next question. Are you able to handle classical reductions like PDC and or LiAlH4?
Yes, we can do both.
Okay, great. That's all the time we have. Thank you, Dr. Yan Xu and Declan Ryan, for that excellent presentation. Thank you, participants, for being a great audience, and be sure to check CNEN or CNN Online for information on the next edition of CNEN webinars. Thank you, On24, for technology and production services. And thank you, WuXi AppTec, for the sponsorship that made this interactive webcast possible. For CNEN webinars, I'm Kelly McSweeney. Goodbye.
Thank you.
Thank you.